the therapeutic aspects of EGFR, PIK3CA, and FGFR. One primarily that we found from this study. We found that these alterations were specifically the EGFR mutations, they were significantly associated than what we normally tend to get to see in the adenocarcinomas. They were associated with the non-smokers and also with the gender type. So we went on to characterize the whole genome, and that's a kind of like a descriptive analysis that what we present. But that's not exactly the kind of like focus for today. The focus is that after characterizing these genome, what we found is that PIK3CA pathway, the FGFR1 pathways, and the EGFR are targetable outcomes also, and can be extended, the treatment that is being offered in adenocarcinomas, to the squamous as well. And now, as a routine practice, possibly that this study kind of emphasizes that even in the lung squamous, these, especially the EGFR, should be genotyped. And we found the mutations, those were the standard mutations that you otherwise normally find in adenocarcinomas. So that was about the adenocarcinomas, taking a shift into, that was about the squamous, taking into the shift. This was another study, one of the first graduate student from my lab, Dr. Pratik Chandrani, who is now a faculty at Tata Memorial, who is also will also be talking about today soon, later. What Dr. Pratik Chandrani did, he took a set of samples of about 400 samples, where he took as a validation, and then again the same similar approach of next generation sequencing platform. And what he found was again interesting. So what he found is that yes, in lung. So again, this is one of the first studies from India characterizing the alterations in underlying the lung adenocarcinomas at a resolution that has not been reported before. So what Pratik finds is that there are significant mutations in EGFR and KRAS, but then Pratik also finds significant alteration in another tyrosine. That was a major discovery that, along with Dr. Kumar Prabhash and I, we were able to take it further, show the clinical relevance of it, and get it published. This was published. This is a published work. So here is you would normally get to see in the textbooks and all the citations about with regards to the Caucasian population, with regards to all the therapeutically relevant alterations. What we get to see is that there is a significant alteration in addition to the alterations that what you otherwise see in the Caucasian population, in the Indian specific, in FGFR3. Now, FGFR3 is a druggable gene. It's a tyrosine kinase again. We initially thought that this might be something which might be restricted only to Indian, but then there were reports of Korean population, again, significantly reporting significant alterations in FGFR3 in lung adenocarcinomas. So the laboratory kind of goes ahead and characterizes these mutations that were derived from the patients. So we take these mutations from the patients and we put it into mice. And what we find is that these mouse, they get they form tumors. And when we orally give them FGFR inhibitors, which are commercially available, we see a significant reduction in the size of the tumors, which is emphasizing that the discovery that was done in the laboratory while characterizing the genomes of these adenocarcinoma patient has some therapeutic implications. So initially we started with doing with the skin xenografts where the tumors were formed in the skin. But then the question that comes up, that Kumar Prabhash and I, we kind of like realized that if we really want to show its clinical relevance, we will have to make the tumors in the lung. And for that, we will have to do more sophisticated transgenic mice, sophisticated mouse, where we will have to do an orthotopic. So another postdoc in my laboratory, Dr. Ashwin Butle, started these experiments, these sophisticated experiments, where the cells were directly injected into the lungs, and here there is a dye that was injected, and that has been shown over here that the injection was precise, and we could see in both the arms. So this is how these cells that what we were injecting, these cells have an luminescence with it, okay, a luciferase. So what happens is, like, when you put them under a camera, you can see that with the growing day, this is how the tumor, the intensity is growing. And then after 30 days, when we go ahead and sacrifice the mice, what we found when we cut it open, what we found is that there were full blown tumors right in the lung, which is very similar to what we kind of get to see in human. In fact, there were like pleural effusions, and when we take these pleural effusions and when we throw it under the bioluminescence, what we see is that these luminescence, that means that these cells are the tumor cells, something similar that what we get to see in the human thing. So that allows us to establish a bona fide lung cancer mouse model in mouse. So again, this is one of the first model, an orthotopic model that allows us to ask questions. For now, we are going ahead and asking the FGFR3 relevant questions directly into the mouse. I mean, these are the histology that kind of like really shows that the tumors that what we get to see in the mouse lung are they are indeed adenocarcinomas. So in this effort to summarize the lung cancer aspect, the efforts that has been done along with Dr. Kumar Prabhash at the Tata Memorial Center in my laboratory, we have characterized the first high resolution lung adenocarcinoma genome, the first lung squamous carcinoma genome. What I did not talk about is the small cell as well, but we will leave it for some other day. I would change gears. I would change gears in the rest of the time. As I said, I'll be giving an overview of different tumor types that what we have been kind of like characterizing, and to show a strong emphasis and initiative in characterizing and understanding and finding targets which are relevant for the Indian patients. So here is the demography of the early stage tongue cancer patients, and this was done in collaboration with Dr. Sudhir Nair, who is an oral cancer surgeon. I would skip this demography part. Again, this is a heat map representation. We take the next generation sequencing platform as to our rescue. What we do is that we take the oral tumors, surgically resected oral tumors, and then we go ahead and we sequence them, and then we develop our in-house algorithms and computational pipelines to analyze that and try and make sense out of it. And this is what is shown over here. Here are the genes, and here are the patients, and here are the mutations in those genes in those patients. And what we found is that again, there is something very interesting as a concept which is kind of coming along from these studies. As what I said for the lung cancer as well, that yes, we are finding mutations in the genes such as KRAS and EGFR in lung cancer. We are finding in the Indian patients as well. However, what comes out as a striking to us is that the frequency of these alterations are distinct from what has been reported in the Caucasian population. For an example, EGFR, which is mutated at around 10 to 15% in the Caucasian, and 35 and higher in the East Asian. In the Indian population, it is around 23%. And 23 to 25%. Similarly, in the KRAS mutations, which is found to be like higher in the Caucasian population, we find it to be lower than the Caucasian population. And so these kind of like balancing act, I mean, whether there is an ethnicity as an underlying reason, whether it is an etiological agent, we really do not know. Again, here in the tobacco-associated tongue cancer, when we characterized, we found a zero incidence of HPV in the tongue cancer, and which allowed us to that these tumors are primarily driven by tobacco. And yes, we did find strong molecular signatures that were associated with tobacco in these patients. So there were a series of discoveries that were like found from performing these high resolution sequencing efforts with the tongue cancer. And I'm not taking you into that, but what I'm just kind of like talking about, we found a significant alteration in a gene that is called as Notch1. It was reported at the time that Notch1 is an oncogene. Now, this is something which is very important that I'm going to say now. So in the Indian context, what did we find is that Notch1 is not only an oncogene, but it also plays as a tumor suppressor in the same tumor in the same cancer. Now that was something which was a new and upcoming concept around that time in the tongue cancer. But now this is well characterized. It is well accepted that the notch, it's known even in the breast cancer, the notch plays as a tumor suppressor as well as an oncogene in a context dependent manner. So we kind of like go ahead. Here is a compilation of different studies in tongue cancer, and this is where our study kind of contributes, and we tabulate. We do a lot of emphasizing on comparing the alterations in the Indian patients, and we compare it across different ethnicities, and there are some startling and interesting findings. So, with regards to the notch, as what I kind of like found, that notch we found as an oncogene in the tongue cancer. Now that had and histochemically, 40% of the tongue cancer, early stage tongue cancer patients were having an upregulation of the notch pathway. And not only that, we found that this upregulation was strongly associated, the notch, not only the notch, the other components as well, with the lymph node metastasis. So you are aware of that there is a occult lymph node metastasis, and there is a huge unmet need to identify those markers that could predict the lymph node metastasis at the time of surgery, so that the neck dissection kind of option could be avoided, could be spared to the patients. So here is one of the option where we kind of like came up with, and we also found that these notch were strongly associated not just with the lymph node but also with the chewing tobacco. We more recently we have found a more stronger. This paper article is under consideration and under review, where we find the work done by another graduate student, Bhaskar Dharawat, and initially by Pavan Upadhyay, where they found MMP10, that is a matrix metalloprotease, and you could see that. We took the samples from the N0 clinical trial. That was a phase 3 clinical trial done by Dr. Anil D. Cruz at Tata Memorial Center. We could retrospectively, thanks to Dr. D. Cruz for extending those samples, we could analyze and we could show that there was an 86% of the lymph node positive guys, they were expressing the MMP10, suggesting that MMP10 could be a very strong biomarker for predicting the lymph node metastasis. We go on to characterize in detail, but I'm not showing you all the functional analysis associated, but I'm showing you a video where we again similar to the tongue cancer or similar to the lung cancer first orthotopic mouse model that we generate here at Tata Memorial. Even for the tongue, we do the similar thing. So here, Dr. Butle, my postdoc in my laboratory, he is injecting the cells directly into the tongue. This is an anesthetized mice. What you see over here is the bulge, and this bulge is going to soon go away. It's going to diffuse, and what you will get to see over the time is that in a matter of about like 30 days, as shown in the lower panel, there is going to be the tumors formed, and this is how the tumors kind of like really look like in the tongue. So now this allows us to really, now again, these cells have these cells have luminescence associated with them. So we could kind of like track them, and we could find whether there is a metastasis associated. So we have addressed that question, but that's not exactly the focus of my talk today. We do find that MMP10 and using the sophisticated mouse model, we are able to show that there is a there is a strong marker like candidate of MMP10 using for lymph node metastasis. So in last 2 minutes, what I would talk about in brief again is the third. I'm changing gears. We talked about lung cancer. I gave a brief overview about oral cancer. These are all mega programs of my laboratory. These are all pillars, verticals. And here is with regards to the gallbladder cancer, it again an early stage gallbladder cancer, along with Dr. Shailesh Srikhande. And what we did over here is again a similar approach. We are using the next generation sequencing platform as our rescue to understand the complexities, to sequence them, and then to analyze the data and see that if there is a therapeutically relevant alteration. What we found here was very significant in our gallbladder analysis. This was again published, where we found significant alterations in ERB2. ORB2 is significantly mutated in and overexpressed in gallbladder cancer. That is what we kind of like showed in this study to a daunting number of 40%. What we also found is that something more relevant is that these ORB2 mutations, they co-occur with the KRAS mutations. You remember the colorectal cancer where the EGFR pathway, when it comes along with the KRAS mutations. Similar to that, what we found is the ORB2 mutations and overexpression were co-occurring with the KRAS mutations. And we go ahead with the series of elegant biochemical and cell-based studies. We go ahead and study the mechanistics of these. But then what to show you what is relevant is that we only show that these mutations that we derive from the patients when we bring on to the to the mice, they form tumors. And when we give afatinib, an approved treatment for, say, lung cancer, when we found that there was a significant. So what it does is that it allows us to extend the treatment from one cancer to the other cancer. Of course, this is subject to a clinical trial that needs to be done. But the clinical algorithm that is routinely being followed in colorectal cancer is something based on this that what we have proposed. In gallbladder cancer. So if the patient is harboring a occurring mutant KRAS, the patient need to be precluded from any of these treatment. So this study in detail goes and identifies the different alleles of KRAS and then characterizes the same into it. In overall, to summarize. The laboratory, this is one of the only effort from India as an individual laboratory wherein we have performed these higher resolution studies of next generation sequencing across around 500 odd tumor types across different, whether it is breast, cervical, gallbladder, lung, tongue, thyroid, and colorectal. I have only talked about today to you is a brief overview of gallbladder, lung, and tongue. But the whole transcriptome sequencing and whole exome sequencing from my laboratory and the discoveries that is being done in collaboration with the clinicians at the Tata Memorial. So what did we talk about today? What I talked about is in the lung cancer, we talked about the discovery of EGFR, PIK3CA, KRAS, and FGFR1, significant alterations in lung squamous that has potential therapeutic relevance in the clinics. Similarly, FGFR3 alteration in adenocarcinoma to the same effect. In head and neck cancer, I gave an overview of Notch and MMP10 as something that could kind of like predict lymph node metastasis. The major part of this is currently under review. And the gallbladder cancer, we talked about the ORB2 alterations, which wherein individuals could be given the EGFR therapy or could be precluded. So, of course, this is kind of like a huge group, and this wouldn't be possible unless we are working along with all the star clinicians here at Tata Memorial, and that's what we do. So, with that, I bring an end. These are the alumni from the laboratory who have all been contributing and working on to it. So, with that, I bring an end. Thank you.